distribution of the chromosomes in the daughter cell, and this results into wrong number of chromosome, which is called as aneuploidy or polysomy. This topic will come in detail in 12th class, so just remember this much. Just remember this much, that if these pair of chromosomes do not separate from each other, then what will happen finally is one of the cell will get extra chromosome and the other cell will get one less chromosome. This is called as aneuploidy. So this is possible, and in human case also there are many examples of disorders of genetics in 12th class. You are going to learn many examples where aneuploidy has taken place, so number of chromosomes have become wrong. So this concept is for 12th class. Now, what is next? See, in the case of mitosis, prophase, metaphase, anaphase, telophase all have to be followed every time there is a mitotic division. But now what happened in meiosis is, because there are two meiosis going on, meiosis first and then meiosis second, and if you try to recollect the whole information, we learned so far is that during prophase, condensation of genetic material takes place, and then nuclear membrane will disappear, nucleolus will disappear, and in telophase they will come back again, if you remember this much. Now imagine for a second a cell undergoing meiosis first. So if you think the cell should go for telophase first, then after in telophase first what will happen? Nucleolus will come, nuclear membrane will come, genetic material will again become loose, and then when it enters into meiosis second, it will enter into prophase second and again start losing its nuclear envelope, again making its nucleolus disappear. So it's a wastage of time and energy for the cell. So generally what happens is, in maximum of the cells undergoing meiosis, you will find telophase first is skipped, because there is no point of causing these events in telophase. Reason, if these events take place, then it will be again removed by prophase second. That's why both of these steps are skipped, and the cell straight away from anaphase first enter into metaphase second. Please be clear, from the anaphase first the cell will straight away enter into metaphase second, because telophase first, whatever happens in telophase first will be again removed by the process of prophase second. So why should a cell waste time and energy? The only thing cell will do now, after entering into anaphase or after finishing the anaphase first, the cell will enter into interkinesis. It's a brief interphase during which the cell will prepare whatever chemicals it needs for another division, and then it will make its centrosome replicate, because you know one thing very well, that by meiosis 4 cells are formed. That means you need more centrosome. So that's what the cell will do in that gap time between anaphase first and metaphase second, and there is no DNA replication at all. So now the anaphase second is just like mitosis, and in this process chromosomes will not separate, but of each chromosome sister chromatids, just like mitosis, will separate, and it is made up of prophase second if it is there, otherwise straight away metaphase second, anaphase second, telophase second. Now I have made another diagram just to explain. So this cell will enter into, see what is the result of meiosis first? 2 cells are formed, haploid number of chromosome, but amount of DNA is double. So now these both cells are going to enter into meiosis second to form 4 cells. Now 4 cells will have haploid number of chromosome and the right amount of DNA, not the double amount, right amount of DNA. Now they are going to separate. These sister chromatids will separate and each one will enter into one cell. So finally sister chromatids will change into daughter chromosomes. I hope this point is clear to you. So now how do you distinguish the meiosis first from the meiosis second? Now let me quickly tell you some points that how meiosis first is different from the second. So first you can easily remember is that meiosis first is reductional is the heterotypic division, and the meiosis second is homotypic division, or you can say reductional division and the equational division. Then the second difference is the number of chromosome is reduced to half in the case of the meiosis first. That means diploid content of the DNA will change into the haploid content. But in the case of meiosis second, the number of chromosomes remain as such. That means haploid will remain haploid. This is haploid set, this is diploid set. So it turns from diploid to haploid and remain from haploid to haploid. Only DNA amount is reduced to become normal. It's not half, reduced to become normal. Now the next thing is the chromosomes remain. Now in this case, chromosome remains in the replicated state. In that case, in the meiosis second, the two chromatids of the replicated chromosome separate. Here the chromatids are remaining together. You can see chromatids have remained together, only the chromosomes have separated. In this case, chromatids are going to separate from each other. Separated. Then the first one, meiosis first, is a complicated and long process compared to the meiosis second, which is a comparatively simple and a short duration division. And please remember one thing, crossing over takes place in meiosis first, and there is nothing like crossing over in the case of meiosis second. I hope these differences, major differences, are clear to you. Now what happened next is the cytokinesis. So after meiosis, please remember there are two methods possible for a cell to undergo cytokinesis. Now what is cytokinesis? If you remember, cytokinesis is the division of the cytoplasm. So if you remember, I have told you some differences. So karyokinesis is the nuclear division, cytokinesis is the cytoplasmic division. First step is always nuclear division, that means karyokinesis. Second step is always the cytokinesis, that means the cytoplasmic division. Please remember, in the karyokinesis there is distribution, equal distribution of the genetic material, and in the case of cytokinesis there will be equal distribution of the cell organelles, that means chloroplast, mitochondria, etc., etc. And please remember, there will be division of nucleus, equal amount in the karyokinesis, and there will be division of cytoplasm in the case of the cytokinesis. Be clear with these things. So now what happened, the next thing is cytokinesis is of two types, successive as well as the simultaneous cytokinesis.